So our next speaker is Dr. Yufeng Gay, who is an associate professor in the Department of Biological Systems Engineering at University of Nebraska-Lincoln, and he'll be talking on machine learning estimation of leaf chlorophyll and water content.
I can use this mic. Okay, good morning. First of all, I would like to thank the organizing committee to invite me to come over here and give me the opportunity to share some of the research that we do in our lab with you. Again, my name is Yufeng Ge, and I'm associate professor from Biological Systems Engineering, University of Nebraska-Lincoln. And Jia Ting is the first author, is the graduate student who do most of the work here. So this picture is all my collaborators. Like I said, I trained as an engineer, but I work very closely with plant scientists. Some of them are plant breeders. Some of them are much more closer to plant breeders than I am. And I really want to lift them up today because, without them, it's not really possible for my research program, because it is them who allow me to, my group, to get into their field and collect the data and collect the sample from their plots. And the other reason that I show this picture is because you'll see we are all relatively young, and we all wanted to live up to 90 years old. So somebody talk about 100 years of history about the corn breeding, right? I was sitting in the and talk about some of the pioneers in the history, and I was sitting in the audience. I was really motivated because I hope, 2070, some of our graduate students can talk about this cohort. I know that Catherine or maybe Jingliang, some of the graduate students are in the audience, no pressure for you. So in the UNL, I direct what my students call the STAR lab. So STAR stands for Sensor Technologies for Agricultural Research. So I just wanted to give you a very quick overview of what we do in our lab. So here is a robotic system that we developed, essentially that can automate the process to measure the leaf properties such as the thickness and the temperature. And down here is a large-scale field phenotyping facility that we built at Nebraska. And if you haven't been there, we encourage you to go. It is about 40 miles away from the Nebraska campus. And we built this essentially for intensive high-throughput phenotyping. And the field is divided into a couple of hundred plots. And then there's a sensor platform in the middle. It's a cable robotic concept that you pull the cables, and then the sensor platform can position itself to anywhere in the field and do intensive phenotyping. And that's the sensor platform there. So here it is a soil sensor that we developed. So we are interested in developing the subsoil properties. Therefore, so we developed a probe that can be attached to a penetrometer that goes deep into the soil and measure the subsoil properties. And I see that tomorrow there's somebody that talk about, including the soil variation would help with the signal to noise of the breeding program. So that is something that we can also contribute to measure the soil property. And down here, that is a sort of an IoT-enabled soil and water sensor soil and plant sensor network that we developed, and we wanted to really measure that soil, plant, and atmosphere continuum in the plot scale, as well as develop some sort of the engineering systems, imaging and edge computing and wireless communication, those kind of technologies. So if you are interested in any of these technologies, please talk to me after the talk. And on the right here is really the hyperspectral system that my lab also devoted a lot of time to conduct research, and that is what I'm going to talk about today, right? So here is a picture of the leaf hyperspectral signal, and from the hyperspectral data that we can get a lot of the relevant leaf properties that might be useful for the plant breeders. And down here I've showed two examples that is from one of our recent study. We look at the prediction of the nitrogen concentration as well as the potassium concentration. So now talk about the leaf hyperspectral data, right? So if you're familiar with the literature, so many leaf canopy properties can be rapidly estimated through hyperspectral remote sensing. So water is one of them, pigments, photosynthetic activities, and then macro and micronutrients, right? Some of the report also talk about like the primary metabolites that can be detected by the hyperspectral data. And it's useful for detecting pests and disease stresses as well. So, and the hyperspectral technology isn't new, right? So it is traditionally used for crop surveillance and management, particularly in the satellite and aerial remote sensing community. And high throughput phenotyping sort of rekindle its use, and you see a lot of mention about, you have handheld devices that breeder wants to carry to the field to take the in-situ measurement, or there's a lot of research how to get the hyperspectral cameras or the spectrometer on the ground vehicles and the drones for the measurement. And this all from this kind of motivation. And acquisition of hyperspectral data can be rapid and non-destructive and low cost. Don't get me wrong that calibration of the hyperspectral device itself in the field is very challenging. But sometimes it's compared to a lot of other data sets, it is rapid and non-destructive and low cost. So, and it also can be imaging and non-imaging. So I'm going to talk about the non-imaging devices primarily today. So there's also a lot of problems with hyperspectral modeling, right? So I would like to direct your attention to the graph here. So you usually start with the field data, right? And then you divide those into two parts, and then you have a training data sets, and then you have a test data sets. And then from the training data, you do cross-validation and optimization in the hope to come up with a model. And then you apply that model to the test data to get your statistics, essentially to show model works well for this independent test, and you can claim model is actually functioning good. Right now, it's only got two problems. One is that because we are relying a whole lot on the machine learning algorithms for the model development these days. So this training data set is a lot. Usually you see like 70% of the training, 30% of the test. So which means that for all these 70% of the data set, you still need to get the label or the reference, and that is expensive. Right? So that's the first problem. And the second problem is, now put that on the test data isn't your ultimate goal. The ultimate goal really is to apply the model for the deployment, right? So it's gonna be probably a very different population or very, very different environment. And this is where you see the model starts to fall apart because their accuracy is not as accurate as your test data, right? So it's really not scalable what we're talking about. So that's essentially the problem that we see. And there are potential solutions. Right, so the first solution is what we call the calibration transfer. Someone has the model, and you suspect that the model may not be very useful for your data sets, but you can come up with a calibration transfer so that can perform well. And then the other one that we look at the literature is use simulated hyperspectral data from RTM. So RTM is a radiative transform model. And essentially, it's a magnetic model about the interaction between the electromagnetic energy and the leaf, right? So simulated data is free, and we think that this might be some of the potential to solve our problem. So I talked to the Jating, the graduate student, and she's the one have some remote sensing background. And I said, well, can you look into this and try to solve this problem, see where it can lead us? Now, and she agreed, right? So, and then we look into, and we are trying to see whether we can utilizing RTM to help us with the model development. So a little bit about radiative transfer model. So it is developed in remote sensing community. So this is another thing that I really liked, right? So when we are stuck in our own discipline, what you do, right? You look out and you kind of look out at other disciplines. There might be solutions out there for you. And so it has been already there for 20 plus years, 30 plus years actually. So mechanistic models to stimulate the interaction between the EM radiation and the plant leaf. So the most famous one is the prospect. It's the most well-known one, the prospect family that stimulates simulates the leaf level reflectance and the transmittance from that visible near infrared and shortwave infrared region, 400 to 2500 nanometer. So the model really can run both ways, right? So you can run the model in the forward mode. Essentially, you specify a bunch of input parameters, and the model is gonna spit out the simulated spectral data. Or you can run the inversion, the backward mode. And essentially, if you have the field spectral data collected and you want to figure out, well, what is the chlorophyll concentration for that spectra, and you can do run the model backwards and trying to figure out what is the chlorophyll concentration. And model inversion is a yield problem. It's a yield problem just because you can have different combinations of the input parameters that have the same spectra, which essentially is a non-unique solution. Okay, so now, particularly for our research, right? So we are trying to see whether we can leverage that RTM model and bring that into our field data sets, trying to improve the modeling and improve essentially the accuracy as well as the robustness of the model, and to see if we can actually reduce the amount of the field data that we need to collect to make this work. So we collected hyperspectral data from 300 sorghum leaves and 560 corn leaves. And so the field data, for these leaves, we also measured these leaf properties, right? So we have the chlorophyll concentration measured, we have the water concentration measured, and the dry matter measured, and the nitrogen content. Now our focus is really the chlorophyll and leaf water thickness. And then half of the data, we kind of said that we're going to just use that for the model development purpose. And then the other half, it's going to be strictly being used for the model evaluation. Now I also want to point out that we use this corn and sorghum that provide us the opportunity to test the cross of the model, whether if we develop a model from the sorghum, does it apply well to the corn, and vice versa, those kind of testing. Right, and then we simulate the data use a prospect model. And so it turns out that the simulation itself is not quite straightforward, because for the prospect that it gets 10 input variables, 4 of those we figure out where it's not really relevant to our study, therefore those are fixed. Now there's 6 that we have to vary them to create the variation, right? So the 6 of those are here. And it doesn't really matter, there's a lot of numbers here, but the number doesn't matter. So now what really matters is the philosophy of how you're gonna generate the input variable, put them in the model, and then simulate the spectra. So essentially, we got 3 settings. So first is that we sample from the uniform distribution, right? So you have a minimum value of those input variable, maximum value, and we assume it's a uniform distribution across, and then we put those into the model and it's simulate the spectra. Now, naturally you think that the uniform isn't, so the Gaussian is bad. Would it be better? So the setting 2 is we independently sampled from a truncated Gaussian distribution. So we still have the minimum and the maximum, but we are using the Gaussian distribution to simulate what the input variable should be, then to put that in the model, right? And then, so now this independent sample, the Gaussian distribution is still not to represent the reality very well, just because all these variables, if you look at the natural leaf, those variables is gonna be correlated somehow, right? So water content is correlated with nitrogen, for an example, right? So they are not completely independent, so to speak. Therefore, we have to come up with the covariance structure of those input parameters, and this is what we do with the setting 3. And we really wanted to see among these 3 settings to generate input variables to simulate the spectral, which mechanism is the best. And then we also look at the different modeling approaches, and this is where the slides get cluttered and a lot of information here. So the first one really is a direct inversion. Like I said, we can just run the model the backward direction. We have the field spectra, and we apply the models to come up with the variable we wanted. Right, and the second one is we can simulate it, and then we can put those simulated data into a machine learning algorithm and train the model to see if we can come up with a better model simply solely dependent on the simulated data. And we simulate about 2,000 data points from here. And the third method is it's the conventional machine learning approach. There is no simulation. We just use the field data, train the model, and then come up with the prediction. And then method 4 is really what is our focus, right? So we wanted to combine the simulated data and the field data, putting them together, and trying to see if this M4 is better than all 3 previous methods. And among this, among the method 4, we also look at some other details. And essentially, because the field data is less, right, and they are more expensive, we have 2,000 simulated data, but we only have 400 field data for the model calibration. Therefore, as we added the field data to the simulated data, we slightly increase them, because the less field data we can be included in this training, the better, because the field data is what's important, right? But that created the imbalance, because we have much more simulated data than the field data, and the model might be overwhelmed by the simulated data. Therefore, so we say, well, maybe we should just replicate all these field data. So we put all those, so we replicate it many times, and trying to achieve a balance between them. And I see that my time is up, so I need to be quick. So essentially, some of the results here, right? So this is the spectral 3 settings of simulation, right? And then the row down here is the principal component of the spectral data, and the gray is simulated, and then those 2 colorful ones are the real data. And essentially, what we are looking for is what is more naturally kind of presented the property of the real data. And through the comparison, we decided that the Gaussian distribution with the covariance structure kind of simulates the field spectrum much better. So that's number 1. And then number 2 is that we're looking at the results of the prediction, right? So again, I will direct you to the CAB, the chlorophyll concentration. We compare 4 different methods, right? So I want to direct your attention to M3 and M4. So root mean square error, the smaller, the better, right? So you can see for the M4, right? So that's the blue line. So it's constantly smaller than the 3 other methods. So that's good. That's what we're looking for. And also when it increase the field sample size, it's the root mean squared error is going to get much smaller. So that's what we want to show. So I'm really running out of the time, so I'm just going through this. And I wanted to kind of, things we learned through this process, right? So hyperspectral data is a tool, and that is not a destination. So when I'm the engineer, so I spend a lot of time just trying to figure out how the instrumentation is going to collect quality data in the field. But then I realized that that is not the final destination for the plant breeders, because unless that you convert those to the stuff that's useful for them, it's the hyperspectral data itself is meaningless, right? So we have to come up with the model. We have to come up with to build that bridge. And the data expensive, machine learning is data hungry. So those 2 are not going very well together with each other. That's where the mechanistic model might provide the free data for us. And so one other thing is other leaf properties such as nitrogen. So when we do this research, we're really trying to address nitrogen, right? But then we figure out, well, there is no nitrogen as an input for those prospect model. So and then we talked to the remote sensing community guy, and they were like, oh, we are still developing it. And that really gets interesting, because that's where I feel like there is not a very clear boundary between physical-based modeling and machine learning-based data-driven modeling. So there's no chicken and egg. It's more of a chicken and eggness between them. So and then finally, our hypothesis is validated, right? So similar field data show higher accuracy, robustness, and we only need a low number of field data for more scalability. So that's it. I'm sorry I'm running out of the time.